Welcome, everyone. Today, we are diving into a fascinating area of computational biology called transcriptomics. If the genome is the blueprint of life, the transcriptome is the construction site in full swing. We are going to explore how cells decide which genes to turn on, how we can measure this activity mathematically, and why this is absolutely crucial for modern medicine. Let us get started. To understand what transcriptomics is, imagine your body's genome is like a massive master recipe book stored safely in a grand library. Every single cell in your body, whether it is a muscle cell or a brain cell, has the exact same recipe book, but they look and act completely differently. Why? Because they are not cooking every recipe at once. Instead, they only copy or transcribe the specific recipes they need for their unique jobs onto little index cards. Transcriptomics is the science of reading all those little index cards at a given moment. Now that we have our analogy, let us look at the formal biological definition. Transcriptomics is the comprehensive, quantitative study of the transcriptome. The transcriptome is simply the complete set of RNA transcripts produced by the genome of an organism at any given time. It is crucial to understand the contrast here. While your genome is relatively static and unchanging throughout your entire life, your transcriptome is highly dynamic. It changes rapidly and constantly in response to environmental conditions, diseases, and different developmental stages of the organism. So, how do we actually measure this dynamic transcriptome? In computational biology, we rely on high-throughput sequencing technologies, most notably RNA-seq. This technology allows us to capture millions of short RNA fragments from a biological sample all at once. But raw fragments are not enough. We then use powerful computational algorithms to map these tiny fragments back to the reference genome, count them, and determine exactly how active each specific gene is. This entire process allows us to build a precise, mathematical snapshot of cellular activity at the exact moment the sample was taken. This dynamic snapshot leads us to a critical question. Why is this so important? Understanding the transcriptome is absolutely critical because it bridges the gap between static genetic code and observable biological traits. If we only looked at DNA, we would not be able to tell a healthy cell apart from a cancerous cell because their core genetic blueprints are often nearly identical. By looking at the RNA transcripts, we can actually see that a cancer cell is inappropriately turning on genes for rapid growth and turning off genes for self-destruction. This field is the backbone of modern personalized medicine. So, we know we need to count these RNA fragments, but we face a major mathematical challenge. To compare gene activity, we cannot just count the raw number of RNA fragments, known as reads. Why? Because longer genes will naturally produce more fragments than shorter genes, even if they have the exact same activity level. Furthermore, we have to account for the total number of reads sequenced in the entire experiment, which can vary wildly between samples. To solve this problem and make fair comparisons, computational biologists use a brilliant normalization metric called transcripts per million. Let us look at the mathematical engine behind this normalization. The transcripts per million formula might look complex, but it is actually very elegant. Conceptually, it first divides the raw reads by the gene length to remove the length bias, giving a rate of reads per kilobase. Then, it divides that rate by the sum of all rates in the sample to find the gene's relative fraction of total activity. Finally, it multiplies by 1 million to scale it. T sub I is the final TPM value. R sub I is the raw read count. L sub I is the gene length in kilobases. If raw reads increase, TPM increases. But if the gene length is larger for the same number of reads, the TPM decreases because those reads are spread over a larger physical area. Now that we have our formula, let us see this with a concrete example. Imagine a tiny genome with only 2 genes. Gene A is 2 kilobases long with 40 reads. Gene B is 4 kilobases long with 20 reads. We want to find the TPM for gene A. First, we normalize by length. Dividing 40 by 2 gives gene A a rate of 20. Dividing 20 by 4 gives gene B a rate of 5. Notice gene B has half the reads, but because it is twice as long, its actual expression rate is 4 times smaller. Next, we sum these rates to get 25. Finally, we divide gene A's rate of 20 by the total of 25 and multiply by 1 million. The final answer is 800,000. This means out of 1 million transcripts, 800,000 belong to gene A. So we have successfully calculated the standardized expression for a single sample. This brings us to our next challenge, comparing different samples. In clinical studies, biologists need to see how many times greater a gene's expression is in a disease state compared to a healthy control. We could just divide the tumor expression by the control expression to get a raw fold change. However, raw fold change can be highly misleading. Why? Because it is asymmetric. If a gene is upregulated, the value scales up to infinity. But if it is downregulated, the value is squished into a tiny fraction between 0 and 1. We need a better mathematical tool. To fix this asymmetry, we introduce the log2 fold change formula. Conceptually, by taking the base 2 logarithm of the raw ratio, we stretch that squished 0 to 1 region into negative infinity, making the scale perfectly symmetric for both upregulated and downregulated genes. In this formula, L represents the log2 fold change. ET is the expression level in the tumor or treatment sample, and EC is the expression level in the healthy control sample. If the gene is overexpressed in the tumor, ET is larger and L becomes a positive number. If the gene is suppressed in the tumor, L becomes a negative number, making it incredibly easy to spot disease-causing behaviors at a glance. Let us apply this formula to a clinical example. We are measuring a specific cancer-related gene. We are given that in healthy control cells, the expression level is 50 TPM. In tumor cells from a patient, the expression level is 400 TPM. We need to quantify exactly how much this gene is overexpressed. First, we find the raw fold change by dividing the tumor expression of 400 by the control expression of 50. This gives us 8, meaning the gene is expressed 8 times higher in the cancer state. Next, we take the base 2 logarithm of 8. Since 2 raised to the power of 3 equals 8, our final answer is 3. This positive value of 3 confirms significant upregulation in the tumor. Now that we have done the math, let us visualize the entire process we just learned. On the far left of this diagram, you see the static double helix of the genome. Moving to the right, this genome produces the transcriptome, represented by those wavy RNA strands. Next, through RNA-seq technology, these strands are shattered and sequenced into millions of short fragments, shown as the colored RNA-seq reads. Finally, using the TPM normalization and mapping algorithms we discussed, these reads are converted into the gene expression levels bar chart on the far right. You can clearly see gene A has a much higher quantified expression than gene B. This diagram perfectly summarizes how we turn biological molecules into actionable mathematical data. We have covered a lot of ground today, so let us review our key takeaways. First, transcriptomics provides a high-resolution, real-time window into the cellular machinery by quantifying RNA transcripts, bridging the gap between static DNA and active biology. Second, we learned that raw data must be normalized. Transcripts per million, or TPM, allows us to fairly compare the activity of genes of different lengths across different samples. Third, metrics like log2 fold change allow us to pinpoint exactly which genes are malfunctioning in disease states by providing a symmetric scale for up and down regulation. Mastering these computational techniques empowers us to decode the dynamic language of the cell and develop life-saving medical interventions. Great job today.